Welcome back to Plantopia. Plantopia is the plant health podcast of the American Phytopathological Society, and I'm your host, Matt Kasson. I'm an associate professor of forest pathology and mycology at West Virginia University. This is the 5th episode of Season 4 of Plantopia, and today it's my distinct pleasure to welcome APS President Dr. Nick Grunwald, research plant pathologist with the Horticultural Crop Diseases and Pest Management Research Unit with the USDA ARS in Corvallis, Oregon. Dr. Grunwald is also a professor in the Department of Botany and Plant Pathology and the Center for Quantitative Life Sciences at Oregon State University. Dr. Grunwald completed both a BS in plant science and a PhD in ecology and plant pathology at UC Davis. He served as a postdoc in the Department of Plant Pathology at Cornell before joining the USDA ARS in Washington State in 2001. In 2004, he moved to Corvallis to begin the job he currently holds with the USDA ARS. His principal research interests include the ecology, genetics, evolution, and management of emerging phytophthora diseases, with a special emphasis on the sudden oak death pathogen Phytophthora rumorum and the Irish famine pathogen Phytophthora infestans. He has published hundreds of peer-reviewed articles on these two important plant pathogens, including co-authoring the 2009 genome sequence of Phytophthora infestans. More recently, he has started working on the development of computational and bioinformatics tools for comparative genomics, genotype by sequencing, population genomics, metabarcoding, metagenomics, and diagnostics based on CRISPR-Cas. Dr. Grunwald has served as associate editor, senior editor, and editor-in-chief for Phytopathology and PhytoFrontiers, editor for Plant Pathology, and currently serves as founding editor-in-chief for CABI, Agriculture and Bioscience. He is a recipient of the 2006 USDA ARS Early Career Scientist of the Year Award, the 2007 APS Syngenta Award, the 2015 APS Ruth Allen Award, recognizing outstanding innovative research contribution that has changed the direction of research in any field of plant pathology, and became APS Fellow in 2016, and American Association for the Advancement of Science Fellow in 2019. Dr. Grunwald, welcome, and thank you for joining us here today.
Thank you, Matt. It's a real pleasure and a great honor to be on Plantopia. I've really listened to some of your podcasts, and they're just awesome.
Yeah, one thing that I've realized in the first 4 months is that I've mostly, so far, only interviewed people in academia. And I think that's not really a balanced picture of the members of our professional society and plant pathologists at large. So I'm really grateful that you're here to talk about your experiences with the USDA. But before we get into that, you've been in Corvallis for 20 plus years, but you certainly weren't raised there. According to my research, you are from Caracas, Venezuela, the largest city in that country, and only a stone's throw from the Caribbean Sea. Is that correct?
That is absolutely correct, yes. I was born in Caracas. I grew up there. I grew up bilingual, speaking German at home. My father's German, my mother's Swiss, but also speaking Spanish with all my friends and in whatever activities I had in school and elsewhere. Yeah, I grew up in Caracas. Caracas is a huge city. Back then it was like 5 million inhabitants. Now it's probably upwards of 12 or 14 million. It's a huge city.
So just slightly bigger than Corvallis.
Slightly bigger, yes.
So what was life like growing up there? And what exposures to nature or agriculture did you have, if any, that may have informed your plan to study agricultural and study plant pathology?
Yeah, growing up in a big city like Caracas, there was very little exposure to agriculture, right? You just got your vegetables at the market, maybe, but where we lived, we were about 5 minutes away from the Avila, which is this mountain that is a park that we can go and hike on. There was wilderness right there, very close to where we lived, and we often went and played there after school. We also, when I was young, my parents took us probably twice a month to beaches, and we camped on the beach. And we did this a lot. We did a lot of snorkeling, windsurfing, things like that. So we really grew up in sandals on the beach or barefoot on the beach quite a bit. But nothing related to agriculture at that point. At one point, my parents looked into maybe buying a farm. We visited a farm. That was about it. And as a kid, I visited, obviously, farms that produce coffee beans or citrus farms, things like that. But that was also a school trip. So it's really very little exposure to farming back then.
Sure. And you mentioned that your parents were German and Swiss. Did they emigrate to Venezuela, or are they born in Venezuela themselves?
No, they emigrated. Yeah. My father is a product and a victim in some sense of the Second World War, right? He was still young, not quite out of high school when the Second World War ended, and he fled from East Germany to West Germany. It's actually quite a story. He really fled with no belongings, just a little slice of bread, and survived. He had to figure out how to cross a river. He tried several times, eventually made it. And it was all word of mouth. How do I cross this river? The guards up there, and they might see you, so you need to wait until they walk the other way. That kind of stuff. And he learned his profession there. And right after the war, German economy was very depressed. And he decided that there was opportunity in Venezuela. And back then, Venezuela was booming, right? The oil industry was going at full steam. It was an exciting place to be. And so he emigrated there. My mother, on the other hand, went there for different reasons. She came from Switzerland. She was there as an opera girl, wanted to learn Spanish, and they met there and stayed there. And my father still lives there.
Okay. Yeah, that was going to be another question I had. As you're growing up there in Caracas, I wondered if there were opportunities there in the city for university study, because clearly you did your bachelor's degree at UC Davis, and I wonder about that journey from Venezuela to California and how that kind of transpired.
Actually, there's some steps in my CV that I don't usually put in an academic CV because it's not necessary, but I took the detour. After graduating from high school, I went straight to Switzerland, and I enrolled as a student at the University of Zurich in business administration. And it was basically a major I chose because I didn't know what I wanted to do in life. So I was enrolled in that for quite a while, and I studied everything, obviously business administration, economics, and I liked it to some degree, but I didn't have passion for it. I also took classes in Japanese, I took classes in philosophy, I took classes in theoretical physics. I was really looking around for what I wanted to do.
Sure.
But I kept going, right? And then around year 3 or 4, I met my future wife, and she enrolled as a student, as an undergraduate student in plant science at TETH to study agronomy, right? At TETH in Zurich, which is another institution right in Zurich. And and obviously we became a couple eventually. So I spent a lot of time with her. In her studies, she needed to do an internship on a farm for 6 months. She needed to actually work on a farm as farm labor, the right hand of a farmer, driving tractors, milking cows, and everything. And I was so fascinated with that. I went there every weekend and I started working on the farm. That's how I really got into farming.
Oh, wow. That's great. I mean, I like those stories because it shows that like our paths into science are typically nonlinear. And it's these kind of what are sometimes perceived as paths of misdirection that lead us to our true calling, right?
Absolutely. So let me take this one step further, Matt. So eventually I dropped out because I was really not very enthusiastic about the major of economics and business administration. And I went into farming. And I got a vocational degree as a farmer, which in Switzerland is typically a 3.5-year degree, but because I had a lot of academic credits, I did it in a 1.5 years, but I basically was trained in all the practical farming. I worked on a farm for 1.5 years and took half a year of winter courses to get a degree as a Swiss farmer, which is a really important vocational degree in their society. And I learned everything from driving the tractor with a hay loader and backing into the barn without bumping into things. So I had exams on driving backwards with a hay loader into the barn, and included everything from maintenance of your machinery, changing oil, to welding, to woodworking, to not just growing crops, but obviously what we all would think of as growing row crops and tree crops and milking cows and feeding
Right.
cows. Yeah.
That's really fascinating. I studied forestry as an undergrad, and I had a similar experience where I worked at a family's forest, and we had to work in the sawmill. I had to grade roads with a manual grader pulled behind a tractor. I had to drive the dump truck with gravel to fill in the roads and take loads of logs to the sawmill, tap trees for maple syrup production. And I really feel like those experiences, like that hard labor, really made me appreciate all the aspects of forestry and forest management a lot more than I could just reading about it, for sure.
So true.
Did you have dual citizenship because of your parents' status?
So I had Swiss citizenship in Venezuela, and I never renounced the citizenship, but right now there's no consulate anywhere outside of Venezuela, so you can't renew that passport. So right now I don't have that citizenship. I don't have an active passport, right? So I don't know where I'm the status there. I used to have German citizenship, but when I naturalized American, I lost that German citizenship because of the German laws. Right. So right now, I'm just American.
And I think these are important conversations to have because for me, growing up in the U.S., born in the U.S. with parents who are, U.S. citizens, it's really easy to navigate job markets and university admission applications because it's built around residents and citizens. But if you fall outside that realm or outside that distinction, it means that you have to improvise in certain situations or follow other routes. And that may mean that there's barriers for certain job opportunities or certain, things like that. We can talk more about that as we get into your current career with the USDA. But we'll come back to your farming. You're a farmer now, but at some point, you go from farming to an undergraduate degree at Davis. Tell me about that next move then.
So after getting my degree in farming, I managed to farm for a year and a half. And then I decided I really wanted to go back to school. And my wife was finishing her studies at DETH in Zurich. And she was going to do her master's thesis in California at a vineyard. And so I decided I might just as well go to UC Davis and study there. Her internship didn't pan out, but I got accepted to UC Davis, went there and finished my undergraduate. Basically, I took all the biology and chemistry, biochem and all that stuff, right? All that scientific background that I didn't get in my previous studies. And finished the undergraduate in, I think it was 5 quarters, so about a year and a half, and then went straight into a PhD.
And was there something particular about Davis or California that compelled you to stay there versus going elsewhere for your PhD?
So Davis is a great place to do a PhD in agriculture, right? It has an incredible breadth and depths of all kinds of disciplines. At the time, I was very interested in sustainable agriculture and organic agriculture. They had a really great program there. They had the student farm, and I was, as an undergraduate, before entering the PhD, I was doing undergrad research in a lab, working at the student farm at Davis and doing research on nutrient management and corn and irrigation and all of that. And I got really fascinated with that. And I decided to just enroll at UC Davis and was accepted into graduate school. The graduate school I applied for was actually ecology. They had this agroecology program.
Right.
So I wanted to stay very interdisciplinary at the time. So very different to what I do now. Back then, my interests were very broad and very much around sustainability.
Yeah, I saw that your degree was ecology, plant pathology. Similarly, I studied for my bachelor's, I studied forest ecosystem science, whereas there was forestry, but there was this other track to get more comprehensive and a little more hard science. But I think it was great to have those other elements that would otherwise be excluded from my study. So what specifically did you work on for your PhD in terms of pathosystems or diseases or pathogens? Was there one in particular that you focused on?
Yes. So I was very interested in soil microbial ecology and soil health. My mentor was Arina van Bruggen. She's now long retired. And She left Davis, worked in Wageningen for a while, and then at the University of Florida, and is now retired. And I think she might be back in the Netherlands. So she had this program where she studied cover crop decomposition. So this is what I did my PhD on, understanding the effect of oat-veg cover crop decomposition over time, on how it changes the soil microbial community, how it changes soil carbon and soil nitrogen cycling, and how that in turn impacts damping of diseases caused by Pisium and Rhizoctonia.
So still working with oomycetes, which became a hallmark of, and still is, the kind of central focus of your career. But really, it was Pythium and Rhizoctonia. And I think I saw that you worked with Sclerotenia at some point in your CV, but definitely Pythium and Rhizoctonia. If you scroll all the way down your Google Scholar, which took me about 3 hours, I got to the bottom. And I saw that there was some publications on Pythium and Rhizoctonia. So I said, that must be what Nick worked on there. So after your PhD, you go to Cornell to do a postdoc. But a lot of that work is actually remote work in Mexico. Is that right? And you want to tell us a little bit about that work? Because it's obviously very interesting. And before I let you answer that, a previous guest of mine, Manon Gallegly, who's a professor here, really told some of the earliest stories of figuring out sexuality and pea infestans, but you clearly took that to the next level with a lot of your work. And I think that kind of built up to this 2009 Nature paper, a lot of other deliverables out of that. But certainly I'd love to hear about your time in central Mexico and what you learned there and what you saw.
Yeah. Yes. I left straight with my family. By then I had 3 kids. I had 3 girls that were born during the PhD. So I call this tongue-in-cheek my most productive period. So I was hired by Bill Fry at Cornell to work in Toluca in Mexico. And I think Manon Gallegly worked there too. And I remember the famous paper by Gallegly and Galindo where they described formation of oospores for Phytophthora infestans from that region in Mexico, right? Mm-hmm. And that is considered the center of origin of potato late blight. So I was really hired to study the biology of Phytophthora infestans, the potato late blight pathogen in central Mexico. And we lived there for 3.5 years. My postdoc lasted a little longer than 3.5 years. It was about 4, maybe 4 and a bit. Most of it in central Mexico. So we lived in Metepec, which is a little town in the Toluca Valley near the INIFAP research station. So this is like the National Research Institute, the Federal Research Institute there. And this was also the place where John Niederhauser did a lot of his famous work that got him the World Food Prize. Mm-hmm. And it's also where CIMMYT used to do a lot of research back when Norman Borlaug was doing research on cereals and corn and bean and potatoes, right? They were working on a couple of crops early on. And John Niederhauser started working on potatoes and got really fascinated with potato late blight.
Right. And Manon shared with me some photos from the 65 potato growers meeting in the Toluca Valley with Niederhauser. And he stayed and worked there with Niederhauser for a summer or two back in the 60s. And it was really interesting to hear some of these stories because I asked him what it was like to work with these scientists who are known for receiving the World Food Prize and doing so much for agriculture and for plant pathology. And to him, it was just, that was my friend, but he's from a different generation.
I had some similar interactions because John Niederhauser was still active and alive back then. So I interacted with him a couple of times, and I actually had the great pleasure of being able to work in the John Niederhauser library, his own personal library, with all his notebooks and all of his research. And there's one paper that I have in phytopathology where I went through some of his data sets for some of the native Criolla type potato varieties. And I published actually some of the data from those studies.
Oh, that's amazing. And to have access to that you must have felt like a kid in a candy shop in a way.
It was a very special postdoc. It was awesome.
After that, you moved to Washington State. Is that right?
Yes, I moved to Prosser, Washington, and this was my first career job. I got a job as a research plant pathologist with the USDA Agricultural Research Service in Prosser, Washington. And my job was to work on cool season food legumes. I worked mostly on fresh peas, but also a little bit on chickpeas and lentils. And the diseases I dealt with were one oomycete, Aphanomyces euteiches, also Fusarium wilt, Fusarium root rot, and a little bit of Sclerotinia work also. Sclerotinia is very important on these centripivot irrigated crops because it's a very moist environment, and Sclerotinia really does well in those environments.
Sure, that makes sense. And you also, so you were primarily a USDA employee during that time, but you also had this adjunct professorship at Washington State University. Is that correct?
That is correct, yeah. Yes, Prosser is about a 3-hour drive from Pullman through the Palouse, and I drove there regular. I really wanted to get involved with the department, so I drove there probably once a month, sometimes more often, tried to go to some seminars and meet with faculties so that, because Prosser is a smaller research station, right, I really wanted to network and develop collaborations with people on campus. So it was really important for me that I was adjunct and active in the department.
I think that brings up a good point and a topic of interest to certainly early career plant pathologists that may be thinking about working for the federal government. I know a lot of USDA folks, whether it be the Forest Service, that also have these adjunct appointments at universities in the same towns where their facilities are. And is that encouraged by USDA to pursue these kinds of professorships, these courtesy appointments? And do you see that as one of the great advantages of federal work, is that you can have a little bit of that academic life without having the crazy life of a purely academic person?
Sure, yeah. So what I see what ARS scientists are doing here at the location of Corvallis where I'm now is that some of them are very active in the department and others don't even participate in departmental activities. I myself am very active. I serve on the Promotion and Tenure Committee. I mentor people. I had graduate students. I have postdocs. I have staff at Oregon State University. I was recently just on a search committee to hire serial pathologists. So I'm very active in the department. I really enjoy that. And I have lots of collaborators in the department. And I agree with you. It's a win. Things are a little bit different in USDA than they are in academia, than they are in the university department. We don't teach. If you don't like teaching, a government job is maybe better suited for you. But some people like to teach or do extension. We don't do much extension. We do some extension. We have to interact with growers. But extension is not part of our core job description, right? We really are hired to be 100% research oriented.
It's my understanding in talking to the few USDA folks that that I've mentored and have gone on for USDA positions, that one of the advantages comes down to being able to actually have grad students versus postdocs. Is the funding, the way the funding is set up, is it harder to have graduate students if you don't have a courtesy or adjunct position with a university? Is my understanding of that correct?
To some degree, yes. For example, when I was in Prosser, because I was 3 hours away from Pullman, it was really hard for me to get graduate students attracted to Prosser, right? They really wanted to be on the main campus. Here in Corvallis, it's very easy for me to get graduate students, and I had many graduate students, both for master's and PhD, and it is very straightforward. Yeah, you just have to be a member of the courtesy. You have a courtesy appointment in the department and in the graduate school. And once you have that, you can mentor graduate students. And in our USDA unit here, I think we have about a third of the students in the department. So the department really appreciates that we mentor so many students. It really adds richness to the department. So it's a win for both sides.
No doubt. And I think every institution I've been at that had a USDA office or facility on campus, there's always been that synergy and that crosstalk. So certainly, I think most people would definitely acknowledge that it is a win situation. Now, you were working for a couple of years in Washington until you applied for and got the position you're in now. Starting in 2004, obviously there was a different job than in Washington, right? Tell us about your job now. Obviously, your role has changed as you've advanced in your own career with the USDA, but what primarily do you focus on with USDA ARS there, and what are you working on currently?
Yeah, so I was hired to work on sudden oak death caused by Phytophthora morum. It was an invasive pathogen that emerged in the mid-90s in California, and around 2000 started causing dieback on tan oaks in southern Oregon. And it also is a pathogen that is not just important in the forest, killing trees, killing oaks, but also in the nursery industry because of federal quarantine. The nursery industry in Oregon was under incredible threat of being subject to these federal quarantines, and that's when I was hired here. So back then, some 20 years ago, if a nursery was found positive, it was quarantined, it was shut down for 3 months, they lost customers, the crops were destroyed. So crops like rhododendron, lilac, viburnum, all those that are susceptible to infection by Phytophthora morum were completely destroyed. So it was a really critical time for the nursery industry. Many nurseries went out of business. It cost the nursery industry millions of dollars. And at the same time, there's this other phase in the forest, right? And in the forest in Oregon, the epidemic is still expanding. The state and the federal government is spending more than $2 million per year just to contain and manage this epidemic.
Oh, wow.
And this is just Oregon, right? And California and Washington also are spending a lot of money working in the nurseries or in the forest in the case of California. Now the situation is a little bit different. In the nursery industry, it seems like the growers are not in the same kind of panic anymore. They see this pathogen as a lower priority. It's just another Phytophthora. They can manage it. And it's basically just following good best management practice. They can make sure that they don't introduce it into their nursery. And if they don't introduce it, then their business goes on as usual. To some degree, the work that many of us did, I've collaborated with many colleagues at Oregon State, Oregon Department of Agriculture, or Oregon Department of Forestry, US Forest Service, and so forth. It seems like for the nursery industry, we have, to some degree, provided solutions that work for them. The epidemic in the forest in Southern Oregon, however, is still ongoing and is a big problem.
There's a couple things that I want to circle back to. The first being that as a forest pathologist, I could really appreciate sudden oak death. And I could also appreciate that Phytophthora morum causes two distinct diseases. It causes a bleeding canker disease of oaks and lithocarpus and tan oaks, and it causes a foliar blight of a lot of these understory plants. But like a lot of major introduced pathogens that have wreaked havoc on our native ecosystems, thinking back to chestnut blight, there's always something good that comes out of it. Maybe with chestnut blight, it was the Plant Quarantine Act of 1912. But I think some of the trace forward programs that came out of the nursery industry unknowingly spreading around P. ramorum, it gave us a lot more clarity about how these plant distribution networks work throughout our country. It's unfortunate that we didn't have a better understanding of that beforehand, but the fact that we have that understanding means that is a less likely pathway for new pathogens because of these kind of safeguards we've now put in place. Go ahead.
I think you're absolutely right, Matt. Phytophthora morum was an example. It's a pathogen that emerged that we didn't even think about, right? We did not think that a Phytophthora would cause that kind of disease. And APHIS was very good at implementing a rapid quarantine and starting with this trace forward and trace backward analysis. And I was involved with them. I have colleagues at APHIS that are co-author. One of my papers published in PLOS Pathogens, where we did a population genetic analysis where we inferred sinks of pathogens and sources of pathogens, right? And the sources were obviously the nursery industry on the West Coast that shipped plant material east to retail locations.
Right.
And the population genetic data completely matched the trace forward, trace backward data of the material, the ornamental plant material that was shipped. So that there's a really good congruence between those different approaches to study that. And I really think very highly of the trace forward and trace backward work, and it's very informative. in understanding the pathways of how we can spread these pathogens.
I agree. And it's my understanding in looking at your body of work is that you applied the same questions to P. ramorum that you did with P. infestans, asking whether or not there was a center for origin. And you found out that was not the case, if I recall. It's slightly different, but it highlighted the importance of doing those studies, that you might think that it would follow a similar narrative to P. infestans, but it was actually different.
Yeah, actually, the situation of ramorum in the U.S. is very similar to the situation of infestans in the U.S. It's basically a turnover or presence of different clonal lineages that were introduced from somewhere. In the case of infestans, we know that most of these come from central Mexico through shipments of either tomatoes or potato tubers or potato plants, right?
Mm-hmm.
In the case of ramorum, we think it came from Asia now. There was a recent interesting paper by a colleague in Europe, by Thomas Jung, indicating that Vietnam and Japan have higher diversity of ramorum, but they're still clonal populations. So my thinking is that we still don't quite know where the center of origin of ramorum is, but with infestans that has been studied a lot longer, we know that there's a center of diversity in Mexico and that populations there are sexual.
Mm-hmm.
But in most of the rest of the world, the populations of Phytophthora infestans are actually clonal.
One of the things that I've been thinking about in recent years, and I've written about this and I've thought about this a lot, is that under a changing climate, the precipitation patterns in the East are different than what they once were. I look at winters now, and here in West Virginia where I'm at, and very seldomly get precipitation in the form of snow in the winter. We're now getting rain, and our soils don't freeze as much, if at all, so we have water-soaked soils. So the moisture regime is changing in an area where we have a lot of susceptible oak species to P. ramorum. They've done the prediction maps, and they don't have really any evidence of natural outbreaks of P. ramorum in the East. But do you think under a changing climate and these changing weather patterns, the likelihood of P. ramorum being able to establish in the East is becoming like more of a reality?
I think so, for various reasons. So with the changing climate, the plants are maybe not as adapted. They might be slightly stressed. And I could really see, if ramorum is introduced, obviously, that's the first step that would need to happen, that it could cause disease on these native forest plants. The other important caveat to this is that I was involved in a study in Canada where colleagues in Canada found a few strains in a Canadian nursery that looked different. And we sequenced those genomes and analyzed those, and it was clear that they were a sexual recombinant between an A1 and an A2 strain. So we could match the genomes of the NA2 lineage and the EU1 lineage, which are of opposite mating types, and saw that those two isolates that we sequenced were clearly hybrid progeny of a sexual reproduction event. In the forest in Oregon now, we have A1 and A2 populations in very close proximity. So I'm really afraid that if sexual reproduction occurs in the forest, we'll have new genotypes, that these eventually could result in new genotypes that might be adaptive to new climates and new ecosystems. So there's yet another dynamic that is emerging with this pathogen.
And as someone who works on emerging pathogens, you could also appreciate the fact that under a changing climate where you have unique plant stresses not seen in years, if not decades, if not centuries, some of these endemic Phytophthora could become opportunistic invaders of uniquely stressed trees and kind of muddy the waters in terms of figuring out the distribution without actually sequencing or isolation. There could be other Phytophtheras that have been characterized in the last 20, 30 years here in the East that basically act opportunistically on our eastern trees and produce similar symptoms.
Yeah, I think you're absolutely right, Matt. I think a good example of this is Phytophthora cinnamomi, which has a northern latitude border where its disease-causing range ends, right? But with climate change, it can move north into new ecosystems. And since it's a broad host range pathogen, the prediction is that it will probably cause disease where it hasn't caused disease yet in new ecosystems and on new plant hosts.
Especially in areas where they once had frozen soil for winter and snow cover, and now they have just saturated soils that allow water molds to do what they do best.
Exactly. And you don't have the winter kill, right? The Cinnamomum is more of a hot temperature pathogen, right? So it can invade new ecosystems that it couldn't live in before.
That's all great. Thank you for your kind of insights into Phytophthora work, and I'm certainly willing to let you share some other things. I just want to get through some of the other questions I have that are relevant here. I think a lot about our professional society, the American Phytopathological Society. You and I are sitting here on an interview for Plantopia, which is the official plant health podcast of the American Phytopathological Society. And I serve as host, not for a paycheck, but because I want to give back to my professional society. And APS has given me a lot, and I've given a lot to them. Obviously, you've been involved with APS for a long time, and you bring commitment to professional society to a whole nother level. Clearly, you're the current president of APS, but you're the former editor-in-chief for phytopathology. You've been chair of publications board and numerous roles. Talk about what got you involved with APS initially and why you kept doing it.
Yeah, Matt, it's a great question. I'm asking that myself. I think there are several reasons for why I love working with APS so much. So probably I got really drawn into APS starting as an editor, even though before I served on some boards and committees, but mostly as a committee member or board member. But I really got groomed or worked myself into leadership positions as an editor. So I started as associate editor for 1 year, then became senior editor for 3 years, then editor-in-chief.
That was all for Phytopath?
That was all phytopathology, yes, at the time, long time ago. And then right after stepping down as editor-in-chief, I was asked to become the chair of Pub Board, of the publications board, which supervises all of the journals. And that was a very critical time for APS because our journals were threatened by the emergence of all the open source, open access journals like PLOS ONE, for example, but also the Frontiers journals, PRJ were all emerging. And in some of our journals, submissions were declining. It was really a little bit of a crisis. So APS established a task force, and I chaired the first task force together with a consultant. We came up with a bunch of strategies, and many things changed after that. We streamlined some of the production pipeline. But more importantly, the big thing is that we positioned ourselves for the open access world. And this was many years ago, right? And now we're still not quite there. We're still transitioning into the open access publishing world. But meanwhile, APS was really good at reacting to it and being proactive. And we launched 2 new open access journals, Phytobiomes and Phytofrontiers. Mm-hmm. They're both doing very well. So that basically, this activity of thinking strategically and long-term and having the resources of APS and the wonderful colleagues that all volunteer their time and are all engaged and all working really hard and pulling in the same direction. It's the kind of environment I wanted to be in, right? So it's very receptive to strategic thinking, very receptive to everybody working together and networking and collaborating and pulling in the same direction. So it's a very wonderful group of people that really love working together and volunteer. Enjoy volunteering.
And I think so often we talk about, and we should talk about, the considerable burdens we put on early career individuals in terms of volunteering for things, not just in professional societies, but in departments and whatnot. And I know myself, I look around a lot when I'm in leadership for my Potomac division, which is my division for APS, and I've been president for that, and I've been forum rep. I don't see a lot of men volunteering for these positions, and I think more need to. And there's just something that I'm always aware of, that you need to use your privilege for good. And clearly there's ways to find the best ways to contribute. You can put your name down as a committee member, but it doesn't mean that you're actually inspired by that work. This work, for me as host, has really opened that up. I like the leadership for the division and things like that. I like talking to people. I like hearing their stories. So it's good to hear your involvement. And it sounds to me that you obviously have published a lot. You think about publishing. You think about the types of deliverables those publications represent. And to ensure that APS was keeping up with kind of changes in publishing and trends in publishing, but also ensuring the journal's standards were high. It sounds like you got involved with that because that was something really important to you.
Yeah, absolutely. The journals are critical. They sustain our society, right? And with the switch to gold open access in our journals, that is just around the corner. We only have a few journals left now that are not gold open access, right? Phytopathology, Plant Disease, MPMI was switched to full open access because the community publishing in MPMI really wanted it. So that revenue is going to decrease drastically because we will lose all the subscriptions from libraries that really provided a lot of income for APS. Yeah, the publications are really what sustains APS and makes it possible for us to have many activities that we can sponsor that we couldn't otherwise. And APS is unique among many scientific societies in having this really solid portfolio of journals that provide an income to sustain the society.
I think the other aspect of publishing that's been a great thing to keep up with kind of commitments to open access is that a lot of journals, and APS is this way, is that they are okay with you pre-printing your stuff before submission. And I think that's also a nice thing because not everybody can afford gold open access, but you still want an accessible version of your data and your paper, and that allows you to do that. But I certainly for me, publishing in phytopathology and plant disease, and I've published each a chapter from my PhD in plant disease, a chapter from my PhD in phytopathology. That felt really good because this is what I'm supposed to be doing. It's getting stuff from my society, but also enjoying the benefit of being able to submit to their journals and publish in their journals.
Yeah, I was just going to follow up on that. When I was PUB board chair, we made APS journals, phytopathology and plant disease, and MPMI, which at that point was not gold open access. We made them green open access, meaning that papers are open access after 1 year. So they're only behind a paywall for 1 year. And we did this strategically so that we could still have this broad recognition and citation, because we know that papers that are open are cited more than papers that are behind a paywall.
And I think that's great because nothing enrages me more than going back to find a paper and it's still paywalled and it's from 1994. And I'm thinking, why are you paywalling a paper from 1994? But yeah, I guess we won't spend too much time talking about that. Let me think of some other things So clearly it sounds like you feel APS is keeping up with the changing publishing environment and making decisions that best meet the needs and demands of its membership, right?
Yeah, absolutely. The current and the past chairs of the publications board are really doing a wonderful job. George Sandin is the current chair. They're really on top of it, and Krishna Subbarao was before him. They're really strategically thinking about what we need to do. And with the Biden administration, there is a clear push now to go up in access, I think by next year. So there are still some changes ahead of us. And we will see how this will all work itself out. It's not completely clear to us. But obviously, you mentioned that authors prefer to pay and to publish in some journals that are more affordable than the fully open access journals where the APC is very high. It might well be that in the near future, the APC is the same for all of the journals from scientific societies, and that might not be a selecting factor anymore.
Right.
But there's still some major changes ahead of us in the very near future.
Yeah, and I recognize that it's a lot easier for an individual to pivot than a professional society that represents thousands of members to pivot. But clearly APS has been thinking about these things and leading efforts to move towards a more sustainable publishing future. Obviously, I don't get to interview the president of the society very often, so I thought I would give you an opportunity to tell us a little bit about either what APS is doing or something exciting that APS is doing that you feel like the members should know about or we'll be hearing more about, and give you a chance, if you want to talk about the annual meeting coming up this summer, to talk about any aspects of that, whatever you'd like to talk about. But I felt like it was really important to give you this space to, as president, to talk about something that's from the top down.
So APS is a wonderful society in that it thinks very strategically. So we just developed a new strategic plan, and a lot of the activities we're developing out of that strategic plan are sometimes changing the way we do business, and we're constantly reinventing ourselves. So that's a wonderful thing. Specifically about the meeting in Memphis, I am very excited about the theme of this year, which I think is very forward-looking. It's to, if I can recall the theme from memory, it's something along the lines of we want to harness the technology revolution to improve agriculture, plant health, and sustainability, right? So what I'm really interested in is in exploring all the technologies, like AI is a buzzword right now and it's everywhere, right? ChatGPT just emerged out of the blue and took us all by surprise. It's not just ChatGPT. We have remote sensing, we have spectral imaging from satellites, from drones, we have robotics in the field, we have precision agriculture, we have the Internet of Things, we have Wi-Fi everywhere through Starlink. So there are incredible opportunities for combining everything. And it's not just in the field or on the combine or on the tractor. It's also in the research lab. It's in the private sector, right? The tools that a plant breeder can have now, whether in the private sector or in the public sector, of using high-throughput phenotyping and high-throughput genomics for improving and accelerating crops. We have novel tools with the CRISPR gene editing system. So I think the palette of technologies that are out there right now, I think we're just at the brink of a complete revolution, maybe at the same scale as the Green Revolution, in accelerating what we can do going forward. So the common thread I see through all of this is that everything is moving much faster. Everything involves big data to some degree, right? Whether it's phenotyping, genotyping, measuring, reacting to how the crop grows, modeling the crop. And so the speakers that we'll have this year all address this topic in the plenary sessions that I'm organizing. In the opening plenary session, I'm very pleased to have Dr. Charity Dean. She's our opening keynote speaker. She was very much involved she was, I think, the assistant director of the public health system in California during the COVID pandemic. And so she understood how pathogen can emerge and cause disease. And she now is the CEO of a new startup company called PHC Global. And she's leveraging AI and machine learning to basically do pandemic preparedness, mostly for medical diseases. And this company provides service for any organization or company to prepare itself for any pandemic that might emerge, right?
Sure.
So this is a perspective that we can use also to manage newly emerging diseases like sudden oak deaths or re-emerging diseases and so forth. And then in another plenary session, I have 3 speakers that will speak to 3 different topics, all related to this theme. One of them develops genomic biosurveillance tools, having probes in the field that can sample spores out of the air, sequence the microbiome, and predict what spores in real time are flying through the air in a given environment. And this is obviously very useful. This is a DARPA-funded project, obviously very useful for surveying human diseases or animal diseases, but it also works for any airborne disease like botrytis in a greenhouse or anywhere else. Another person will be speaking on high-throughput phenotyping and developing tools, computational tools, for analyzing and managing the high-throughput phenotypic data
Actually, you would talk on that for a second, because one of the things that I noticed in looking over your own work is, and I really appreciate about your work, is that to really study the populations of organisms that you look at, and a lot of plant pathologists look at, we're now dealing with so much data, terabytes of sequence data. And I really appreciate that you are developing tools for R and other bioinformatic tools to better dig through this complex ecosystem of data. I think right now it's become really affordable to sequence really high coverage genomes, whether we're talking short read, longer read sequencing is a little more pricey, but you can quickly generate sequence data, but processing it and analyzing it in a way that's meaningful is really important. I thought I'd give you a chance to talk about like some of those kinds of like R-based tools that you're developing and the importance of keeping up on kind of bioinformatic tools to be a better rounded plant pathologist.
You absolutely hit the nail on the head. When I started doing population genetic work, I used something like 10 different programs and had to create something like 60 different data sets for the whole population, for each of the subpopulations, for each of the subpopulations. And then if I made one mistake, I had to correct it in all 60, 70 data sets. The analysis would take 6 to 9 months, maybe 12 months, even if it was a complex data set. Out of that, we wrote an R package called Popper. I had a really nice graduate student, Jan Kamvar, who led that effort. He just did a tremendous job. And we basically automated everything so that you have just one input data set, and then you can subsample that data set for each of the subpopulations. And you do one script that analyzes the whole hierarchy of all the sub-data sets that you have in there, right? Mm-hmm. In addition, we added on tools that weren't available for analysis of clonal populations, which I think is one of the reasons that this was used so much. And I think the trick for the computational tools that we developed was that we developed them coming out of the biology lab, right? We had biological questions. I was analyzing populations of, for example, Phytophthora morum. We wanted to accelerate that. We wrote scripts, and then we said, hey, everybody else could use these scripts. Let's just package these into an R package so that everybody else can do these. And then we gave workshops at APS so that people would start using them. And it was funny for a while because I would walk through postings at APS and I could exactly see which figures were created with our package, which was really funny. So that was maybe 15 years ago, right? Now we're working even more so in generalizing this. So we're working on a pipeline that integrates all the analysis of whole genome sequence data. We're calling this package pathogen surveillance. It's in this toolbox called Nextflow that can run on any Linux server, and an Amazon server, an AWS or Google Cloud, or a Microsoft's Azure server, or in any Linux machine on your local cluster at your university. Mm-hmm. And basically, you can throw in one genome sequence or 300 genome sequences. You can throw in a nematode and a fungus and a bacterium, doesn't matter. And the pipeline takes that data, runs all the way through, and gives you a phylogenetic tree of placement for each of the unknowns. And the wonderful thing is, unlike PCR, it really does work on an unknown. It will tell you what genus it is. If the genus is known, it will tell you what species it is. The species is known. But you can give you a population genetic placement in a minimum spanning network or in a tree, if we know it. And all of it is automated. So we're just about to release the first version of this. We're trying to give a workshop also on where we sequence with nanopore bacteria in the morning, and in the afternoon we use our pipeline to analyze that data in a single workshop in one day. We can sequence and analyze the genome. This is something that 10 years ago we could not even have dreamed of.
Yeah, that's great. And I think that's a really good proof of concept to be able to do it within a single workshop. But great stuff. And I think a lot of my graduate students and a lot of early career scientists, they're so much more proficient at coding because they have to be, because the data necessitates that. But it's great that leaders like yourself, who have really done so much for the field, continue to reinvent yourself in a lot of ways because you're hungry for answers, but you're also realizing that these tools, which are expensive and timely to develop, are critical for helping our field move forward, not just our labs move forward.
Absolutely. I think you're very right. It is a lot more effort. It's not just as simple as writing a few functions and putting it out there, right? When you write an R package or the pathogen surveillance pipeline in Nextflow that we're developing, you need to do a lot of quality control because they're naive users that will just throw something in and you get data out the other way. So we need to have QC in there, quality control, to make sure that the genome has enough sequencing depth, that the sequence reads of high quality, that it's not, contaminated sample. So all these little things that we would normally not have to check for if we do it just for ourselves in the lab, right? All these kind of borderline events that can happen, right? These edge cases, you have to program into these tools. So it's a lot of work to maintain and program these tools that a lot of people don't appreciate.
Absolutely.
To give some credit to all the people that developed those in my lab, this wonderful group of postdocs and graduate students that have come through the lab and are still working on these tools in the lab.
Definitely. And I think the thing that I think a lot of PIs think about and is continuing to try to hire people that have different skill sets than us, but that are also more talented than we are, because we're not going to grow if we're the smartest person in the room or we're hiring people that have the same skill sets that we do. We need people that are thinking differently than us and have a different perspective to allow us to come up with great ideas. We're just about out of time, but I wanted to give you the microphone because you're the president of our professional society. There's a lot of people that listen to this podcast that are not part of our society, but are interested and enthusiastic about plants. And some of those might even be high schoolers that are interested in pursuing a career in plant sciences or plant pathology. Any encouraging words for younger listeners out there that might be considering a career in plant pathology and really how to get started, or why plant pathology is so important at this time in our civilization?
Yeah, thank you, Matt. I think plant pathology is an incredibly rewarding and important discipline. People or society does not realize how many problems we have solved and why it appears to everybody that we have a constant supply of high quality, nutritious food. Plants are what really make the planet run. There wouldn't be animals, there wouldn't be clean air. All the nutrients that we consume come from plants. So plants really produce most of the calories that run the world and feed the human population. So plants are incredibly important. As humans, we are creating a dynamic that is not so good, right? We are responsible for climate change, for what some scientists call the Anthropocene, right? For rapid land use change. And I think we need to be careful in making sure that we reverse some of the climate change dynamics. And as part of that, plant health will always have a very important role. We need to safeguard our ecosystems, our landscapes, and our agricultural crops to be able to feed the world. By 2050, we'll add maybe another 2 billion people to this world, to this planet. And there will be a huge pressure to have highly nutritious food and food safety to feed that many billion people. So I think this is a very exciting time to go into agriculture, and particularly into plant health. I also think that we have a whole set of new tools that we didn't have available when you and I went to graduate school. Tools like gene editing and the high-throughput data and the computational tools we have now, and the robotic tools for high-throughput phenotyping or spectral imaging and so forth. So there's a whole set of new tools that we can harness to improve plant health. I think it's a very exciting time to go into plant health.
Yeah, at the same time, I think it's important for those young listeners to recognize that, or realize that we still work with the organisms themselves, and it's important to be able to grow crops and identify pathogens in the field and culture them in the lab. If you're not a data scientist or hungry for bioinformatics work, there's still a place for you in our profession. It's been wonderful talking to you, Nick, and I really appreciate you taking the time to chat with me today about your career and kind of your vision and APS's vision for the future. So thank you so much.
Yeah, Matt, I want to also thank you for hosting Plantopia. You are doing a wonderful service. And I want to also thank Jim Bradine and David Gadouri who started the podcast before you. I think that you're providing a tremendous service to all of us. And thank you very much for hosting me here today. It was a pleasure talking to you.
We've just heard from APS President Dr. Nick Grunwald, Research Plant Pathologist with the Horticultural, Crops, Diseases, and Pest Management Research Unit with the USDA ARS in Corvallis, Oregon, and Professor in the Department of Botany and Plant Pathology at Oregon State University. As always, be sure to check out the show notes for this episode immediately following the episode description. I'm Matt Kasson, host of Plantopia. Thanks for listening.